Welcome students. We were studying this transcription, how this transcription process takes place. What is transcription? That is a segment of DNA will be transcribed or will be copied into RNA. That process is called as transcription. So, in this transcription only one strand of DNA is will be making a copy. Only one strand of DNA will be making the copy. So, that one strand is which makes the copy is the template strand, and the other strand which does not make any copy, which does not code for anything, that is called as the coding strand. Okay. So, here now coding strand will have the same sequence as that of RNA. Coding strand will have the same sequence as that of newly formed RNA. Let us see this one. So, for example, here let me tell you, this is the one strand of DNA and this is the other strand of DNA, two strands of DNA. So, this out of these two strands, one strand is called as the template strand and other strand is called as the coding strand. DNA is double stranded structure. Okay. So, in this double stranded structure so two strands are there. So, one strand is called as template strand and another strand is called as the coding strand. Another strand is called as the coding strand. So, here if this is the template strand, so on the strands nucleotide sequences will be present. On the strands nucleotide sequences will be present. Suppose for example, nucleotide sequence is there. Here A, T, G, C, A, G, C, T, A, T, G. This is the sequence on the template strand. Okay. Now, on the coding strand, you will be having the same. If A is there now what will be there? T will be there here. If T is there, A will be here. If G is there, C will be here. If C is there, G will be here. Now, A is there, T will be here. A is there, again T will be here. If G is there, C. Okay. If C is there, G will be there. T, A, T, A, C. So, this sequence will be there in the DNA. Two strands of DNA, both will be complementary to each other. Both will be complementary to each other. Suppose this is one strand. Okay. This is the black one is the template strand. On this strand the sequence is there. Okay. Now, this is the coding strand. So, this will be a coding strand and this will be a template strand. Let black be the template strand, coding strand. On the template strand you will have this codings. So, I will write this A, then T, A, T, then we have G, then we have C, then we have A, like this coding will be there. Now, on this coding strand also it will be there. If A is there coding strand will have T. If T is there coding strand will have A. If C is there, coding strand will have G. If C is there, coding strand will have G. So, like the same sequence will be there on the coding strand. So, here these two strands of DNA, here it is in the coil form, I have drawn this in the straight, instead of this coil I have drawn straight. So, two strands of DNA, out of two strands, one strand is the template strand and another is the coding strand. Okay. The strands which codes for RNA. Now, out of these strands, one will be helping in the formation or synthesis of DNA, one will not help. Which will not help? This coding strand will not help. This template will help in the synthesis of RNA. So, if RNA strand is synthesized with the help of this template, if RNA strand is synthesized, okay. So, what will be the sequence? What will be the sequence? So, if A is there, then RNA. So, if A is there, the sequence will be U. If T is there, the sequence will be A. If G is there, the sequence will be C. If C is there, G. If A is there, U. Okay, if A is there, again U. If J is there, C. If C is there, G. If T is there, A. If A is there, then T. If T is there, not T, this will be U. Okay, if T is there, A. If G is there, C. Why I am taking U instead of T? Because in RNA, thymine will be replaced by uracil. In RNA, thymine will be replaced by uracil. So, instead of A for T, what I am taking is U. Here, thymine will be replaced by uracil. And what I have said, the new RNA strand will have exactly the same sequence as that of the coding strand. Coding strand is the previous strand, okay. Now, this is the new strand which has formed from the single strand. Only one strand is helping in the synthesis of RNA. So, with the help of this strand, a complementary RNA strand has been made, okay. And this RNA strand, the sequence on the RNA strand is exactly same to that of the coding strand. Is it exactly the same? See here, here it is T, here is also T, but here T is replaced by U, then the same, then A, C, C, G, G. Here T is there instead of T because it is RNA, na? The T will be replaced by U. Then again T is replaced by U. C means G will be there. G will be there. G is, yes, not sorry. So, C, C, G, G, A, T instead of T we have this U. Instead of the A, then A again, C, then C again. The same sequence is on the, that is the sequence which is there on the coding strand. The same sequence is in the RNA. Just the difference is instead of T, it is replaced, the T is replaced by U because it is RNA. So, in RNA we do not have thymine. In RNA, thymine is replaced by uracil. So, apart from that replacement, everything is the same. Everything is the same. Both the coding strand and the newly synthesized RNA strand, the coding on both the strands are the same. Okay. So, what is transcription? The process by which a RNA is made from DNA. The process by which RNA is made from DNA. That process is called as transcription. So, in this RNA is made from where? From how they? From this DNA. From this DNA, RNA is synthesized. So, this process is called as transcription. This process is called as transcription. For the transcription to take place, transcription unit will be there. There will be a unit of transcription. So, what is the unit of transcription? It is the segment of DNA. It is the segment of DNA which is transcribed into RNA. Transcription is nothing, transcription is unit is nothing but it is the segment on DNA. It is the segment on DNA which is which is transcribed into RNA. So, this unit consists of 3 regions. This transcription unit consists of 3 regions. The first is promoter, the second is the structural gene and the third is the terminator. Transcription unit, what is this transcription unit? It is just nothing but a segment of DNA. This transcription unit is nothing but a segment of DNA which is helping in the synthesis of RNA. Whole DNA will not help, only a part. I said you the whole DNA, both the strands are not helping, only one part is helping in the synthesis of RNA. So, that segment which helps in transcribing the DNA into RNA, that place, that region is called as transcription unit. Okay, and that region consists, that unit consists of 3 regions. Transcription unit consists of 3 regions. The first is the promoter, the second is the structural gene and the third is the terminator. Okay. Now, this is the double stranded DNA. This is the double stranded DNA. Okay. So, now the I'm not saying, I'm saying that only a segment of DNA will help in transcription. Okay, so the DNA from here, so here the segment of DNA, that is this segment from here till here, only this segment of DNA is helping in the transcription. This is not helping, this DNA is not helping, this DNA is not helping. Only this section of DNA, that is from this place, only this section of DNA is helping in the process of transcription. So this segment of DNA, which is transcribing into RNA, so this segment is called as transcription unit. Okay, this is called as transcription unit. So in transcription unit we have three regions. The first is the promoter. Promoter, here at the start one blue box is there. I have indicated there won't be any blue box. In order to show you people, I have drawn this one. So blue box is there. So this is the place from where the transcription starts. So the place from where the transcription starts, so this is called as the promoter. This is called as the promoter, the start of transcription, the place from where the transcription is starting. So that place is called as promoter. I'll write here, this is the promoter. Okay, and the place where it ends, here also I have indicated with the box. So place where it ends, it is called as terminator region. Okay, the place from where it starts, it is the promoter region, and the place where it ends is called as the terminator region. And in between this promoter and terminator, that is the segment of DNA which is in between this promoter and terminator. So this part of the DNA is called as structural gene. This part of the DNA which is in between the promoter and the terminator. So this part is called as structural gene. So a transcription unit is nothing but that segment of DNA which is helping in the transcription. Okay, whole DNA will not help, only a segment of DNA will help in transcription. So that segment which helps in transcription is called as transcription unit, and it consists of three regions. The first is the promoter, the second is the structural gene, and the third is the terminator. Let us see what is this promoter, structural gene and terminator, what are the functions. Okay, here first one is the promoter. The first region is promoter. Okay, so here promoter, it is the region of DNA, this place of DNA, because it is starting from here now. So the place where initiation is taking place, that is the promoter. So promoter is nothing but region of DNA that initiates. Initiate means start, that starts, that helps in starting of transcription. So the place from where the transcription starts, it starts from here. It starts from this DNA. See here, this one. It starts from here. So this place from where the DNA transcription initiates or starts, that place is called as promoter region. That place is called, or that region is called as the promoter. And here what is happening is RNA polymerase enzyme is helping in the transcription process. Like for replication of DNA polymerase was the enzyme. I have already said you in the DNA replication process that enzymes plays a very important role. Enzymes plays a very important role in replication. In the same manner, here also enzyme plays an important role in the transcription process. So which enzyme helps in this one? The enzyme called as RNA polymerase. In this transcription, Which enzyme helps? The RNA polymerase. So RNA polymerase will bind at the promoter site or promoter region. RNA polymerase will bind at the promoter region. So this promoter region is the binding site, the place where RNA polymerase comes and binds with the DNA. The place where RNA polymerase and comes and binds with the DNA, that place or that region is nothing but promoter. Okay. So promoter, the region from where transcription starts. The place on the or the segment of DNA from where the transcription starts. So that segment of DNA from where the transcription is starting. So that segment of DNA is called as the promoter. Okay. And one more important thing about promoter, it is actually the binding site of the polymerase enzyme. Which polymerase? RNA polymerase enzyme binds at the promoter region. Okay. And here this promoter is located upstream. Promoter is located upstream towards 5' end of the structural gene. Upstream, downstream. Okay. Upstream and downstream, up and down. So here we say it as there's two locations, one is upstream and another is downstream. So here I'll remove this one, this hole. Instead of this that is helical structure, I'll draw the straight. Okay. This is one strand, this is another strand. This is one strand of DNA, this is another strand of DNA. Okay. Two strands of DNA. This is one strand, okay, and this is another strand, two strands of DNA. Okay. The segment, for example, the transcription is starting from here. Okay. This region is the promoter and transcription stops here. Transcription stops here. This is the terminator region, and this place in between this terminator and promoter, this is the structural gene. Okay. This is the structural gene. So this same, instead of this coil structure, I have put it in the straight structure. So here now this strand, this is the template strand. This one is the template strand and this is the coding strand. Because out of two strands only when one will help. Okay. So this template strand is from 3' to 5' and this coding strand is from 5' to 3'. Okay. So when we show the locations, that where is promoter present, where is the terminator present, where is the structural gene present, we will tell it in accordance with this coding strand. We will take it in accordance with the coding strand. Okay. So here now we said, okay, this is the place, this is the promoter. At this place now the RNA polymerase will bind. RNA polymerase will come. RNA polymerase is the enzyme. The enzyme will come and bind on this part of DNA, promoter region of DNA. So this is the binding site of RNA polymerase. Now here it is located upstream towards the 5' end of the structural gene. So structural gene, which is nothing but we can even call it as coding strand. Coding strand. I said you the reference is from the coding strand. Okay. So now where it is located, where is this promoter located? At the 5' end of the coding strand. So 5' end of the coding strand is here. So where now at this 5' end of the coding strand, it is the promoter. Here is the promoter, that is upward stream. Upward stream of this 5 prime coding strand. So where is the promoter? At upward stream. Okay, promoter is located upstreams towards 5 prime end of the structural gene. Okay, now here the segment, coming to this next one, which is the structural gene. So a part of the DNA or gene that codes for RNA. So from here starting is there. Now here this place in between codes will be there. I said you codes will be there. According to that codes, a new RNA will be synthesized. According to that codes on this, for example, if we take black as the template strand, so on this template strand codings will be there. On this template strand codings will be there. So how according to this codings, so here a new RNA template strand, RNA strand will be synthesized. Okay, RNA strand will be synthesized. So part of the DNA or the gene that codes for RNA. So part of the DNA which has the codes. According to these codes, RNA will be synthesized. RNA will be synthesized. What codes? For example, A, T, C, G, A, G, C, T. So according to these codes which are on the structural gene, new strand will be synthesized. For example, if I am drawing the new strand here, okay, now A will have U, T will have A, C will have G, G will have C, A will have U. So according to the codes on the structural gene, so according to this codes, new RNA will be synthesized. New RNA will be synthesized. So that was the structural gene. And the next is region of terminator. It is the region which defines the end of the transcription. Now transcription will be taking place of this part of the DNA, only till this part of the DNA. Till this part is over. Now this indicates, this terminator indicates that transcription has to stop now. Transcription has to stop now. So it is located where? In reference to this coding strand, terminator is at downstream. Terminator is at downstream and 3 prime end of the coding strand. 3 prime end of the coding strand or the structural gene, one and the same. So this was about the transcription unit. Transcription unit consists of 3 regions: promoter region, structural gene and the terminator region. The same thing. This is the promoter, this is the structural gene and this is the terminator. Now we will study the process in the next video.